what are different ways by which these raw materials for gluconeogenesis can be synthesized, or how they actually participate. So if I ask you, just recapitulation of last class, because it is very important. If I ask you, name the key enzymes of gluconeogenesis, can anybody name them? See from the copy and tell me, but at least read. Pyruvate carboxylase, phosphoryl pyruvate carboxykinase, that is Pepsi K. Yes, and fructose 1,6-bisphosphatase, glucose 6-phosphatase. Repeat. Pyruvate carboxylase, phosphoryl pyruvate carboxykinase, fructose 1,6-bisphosphatase, glucose 6-phosphatase. Okay. Now, see, read this slide. What do you understand? What? So, in the gluconeogenesis, how many ATPs are required? 4 plus 2, it produces 2 GDP, 6. 4 2 NADH means 6. How many? 16. 6 plus 4, 12. Here, how many are being produced? 2 pyruvate means, okay, forget it. This is 6 plus 2, 8. So, there is deficiency of how many phosphates? 4. So, if we oxidize glucose by glycolysis, we get suppose X number of phosphate, but when we synthesize glucose from the same pathway, by reversing the same pathway, we have to spend more energy. So, will you work in a place where whatever salary they give, it is exhausted just for traveling to those places? No. Suppose you work in Kashmir and daily you commute from Srinagar to Kolkata by flight, and you get a salary of 2 lakhs. So, how many days you can travel in a month? If the cost is 20,000 per day, 10 days. So, if you work for 20 days, then you have to spend 3 times that of your salary. So, will you work there? But how come nature is sustaining this particular step where the production of phosphate energy is less than the requirement of energy? Why? So, this is called futile cycle, a cycle which produces no energy. Those cycles are called futile cycle. Futile means which has no fruit, not fruitful, correct? So, why this? Why do you think this is important? Explain why this cycle of gluconeogenesis takes place in spite of being a futile cycle. It takes place every night. If it is not there, we won't survive. আমরা মরে যাব, এত বড় হতে পারবো না, ছোট বয়সে বাচ্চা বয়সে মরে যাব। বাবা হচ্ছে, it depends on 2 things. Number 1, do we have all tissues who can utilize, or we have the tissues who can utilize only glucose in our body and nothing else? Is there any tissue which can utilize only glucose and nothing else? Bolo. Mature RBC, erythrocyte, which has no other option. What is the other organ which utilizes maximum amount of glucose? Brain. And brain prefers glucose first. If glucose is not available, then what is the next important fuel? Ketone bodies. So brain is very, very clever. When there is glucose in the blood, it utilizes only glucose and nothing else. And when glucose is absent, only ketone body is present, it is the first organ to completely change to preference of ketone body. Then glucose won't see anything else other than ketone body. Okay. Just like the government officials, when government changes, overnight they changes from red to green, green to blue, blue to blue to whatever. Like see, if I tell you, hey, I won't allow anyone to sit in the examination, then everyone will say, sir, how beautifully you teach. The moment I will say everyone will pass, you don't worry, next day no one will come to my class. So when you have compulsion, it changes your preferences. So brain is very, very clever in that sense. He senses very beginning that his body is going to change his fuel molecule. So better we shift to ketone body. But alas, for erythrocyte there is no option, because to oxidize ketone body you need mitochondria, right? And erythrocyte has no mitochondria. So what he will do? He has to depend on only glucose. To supply that glucose, small amount of gluconeogenesis is always necessary in our body, otherwise RBCs will die. Clear? That is why we must operate gluconeogenesis even though it is a futile cycle, otherwise we will suffer from hypoxia. These are all actually regulation of gluconeogenesis, which is not necessary for your syllabus. Don't worry. আমি এগুলো সব তোমায় বলে দিয়েছি আগে, দরকার নেই, এগুলি দরকার নেই। This is important. Now, to synthesize glucose from non-carbohydrate sources, that means amino acids, breakdown form of the proteins. The proteins are present in muscle, right? And site for gluconeogenesis is liver. So from this muscle, large muscles are the most responsible for production of glucose. So how they will run the cycle? They have to supply the raw materials, either lactate or alanine. So there are two cycles. One is called Cori cycle, another is called alanine cycle. One is called Cori cycle, another is called alanine cycle. Now, what is Cori cycle and what is alanine cycle? Both operates, remember very carefully, listen, understand it. It's very, very important. Cori cycle and alanine cycle both operates between liver and skeletal muscle. I repeat, between liver and skeletal muscle. Both produces glucose from pyruvate, correct? But the source for production of pyruvate in liver is different in these two different cycles. For Cori cycle, the source for pyruvate is lactate. For alanine cycle, the source for pyruvate is alanine. Correct? Pyruvate, lactate is converted to pyruvate by lactate dehydrogenase in liver. Alanine is converted to pyruvate by alanine transaminase. Where do you find transaminase enzyme? Mostly in liver. Correct? Now, what is the difference and why do they operate simultaneously, or is there any variation in time? Do you have any idea? Anyone can put some light? Some hints are given here. Cori cycle operates between exercising muscle and liver. Alanine cycle operates between resting muscle and liver. So, when we are sleeping, that time alanine cycle is taking place, and when we are running, walking, Cori cycle is. But how much we have to walk? We have to walk very fast, we have to run, we have to do rigorous exercise, then only it will produce huge amount of lactate. Remember that lactate should surplus, that amount of lactate produced during exercise should surplus the amount of lactate can be transported back to liver by blood. Then only it will trigger Cori cycle. Correct? Right. So, we will see the cycles. Cori cycle, liver, blood, muscle. Muscle glucose gets oxidized to form pyruvate, pyruvate to lactate. Lactate comes to liver via blood. Then lactate converted to pyruvate. Pyruvate is converted to glucose. Then this glucose is again poured into the circulation from the liver. So, it goes to muscle again. Again it undergoes this oxidation. Why this oxidation? Because we are exercising rigorously. Rigorously, not brisk walking. We are walking very fast, putting pressure while doing exercise, testing our limits. Correct? Right. So, glucose may travel back to muscle to fuel glycolysis. Fuel glycolysis. Fine. Hello? So, this is Cori cycle operates between exercising muscle and liver. Lactate to glucose, glucose to lactate. Now, what happens here? First of all, when we are sleeping, we are not exercising, so muscle is relaxing, correct? But slowly our liver glycogen is getting exhausted. By the midnight, all the glycogen stored in our liver got used up. So now our body is sensing effect of hypoglycemia. The moment it comes to the action, the sense comes to the body triggers gluconeogenesis. It starts producing the more and more enzymes which are required for gluconeogenesis. Correct? The protein is broken down. Why? In absence of insulin, protein breakdown becomes more prominent in absence of insulin, not presence of glucagon. In absence of insulin, why insulin is absent? Because blood glucose level is low. So, in absence of insulin, protein breakdown becomes prominent. Insulin promotes protein synthesis. So, insulin absence will not promote protein synthesis. So, rather protein breakdown will get upper hand. Correct? So, breakdown of protein will produce alanine. Many amino acid, alanine is also one of them. Alanine can be transported to liver. It can be transaminated by ALT, alanine transaminase, to form pyruvate, pyruvate to glucose by gluconeogenesis. Right? That glucose is poured into your blood and travels back to muscle. What muscle is doing? Relaxing. It need not operate anaerobic glycolysis. When muscle need to operate anaerobic glycolysis, when muscle is exercising, so it is relaxing, so it is producing aerobic glycolytic product, pyruvate. So pyruvate gets converted to alanine by ALT again, same enzyme, opposite reaction, and this alanine again goes back to here. Right? This is how the cycle goes on. The moment we wake up in the morning and start taking the first bite of the food, insulin start increasing. In presence of insulin, things gets changed. Okay, fine. So this is alanine cycle and Cori cycle difference. Bond kar. Close করাটা হ্যাঁ হচ্ছে না মাউজে কর ক্লোজ কর হ্যাঁ এটা কর হ্যাঁ এটা হ্যাঁ পাশেরটা এইটা পাশেরটা হ্যাঁ So now we will start glycogen metabolism. This is how it looks under microscope when it is stained, taken from a liver slice. Slice of a liver tissue is taken. Inside the hepatocyte we find these large vacuoles containing, see, dot. These are vacuoles of hepatocytes. This is liver cell. This is one liver cell. The demarcation of one liver cell. Okay, this is the cell membrane. And inside there is a vacuole, and these dots are glycogen molecules. Correct? What is the difference between glycogen stored in the muscle and liver? Quickly. এই ছেলে বল সামনে আছিস তুই হ্যাঁ তুই বল Next, what is the difference between glycogen present in muscle and liver? হয় না, লিভারে হয়। So liver can contribute to blood glucose level, liver glycogen can contribute to blood glucose level, muscle glycogen cannot contribute to blood glucose level. Glycogen synthesis and breakdown, these two processes we will see. The process of synthesis of glycogen is called glycogenesis. The process of breakdown of glycogen is called glycogenolysis. So we will first see how a glycogen molecule can be broken down. A glycogen molecule exists in this manner. Like this. Correct? So this glycogen molecule has two ends. This is one end and these are other ends. This end is reducing end and these are non-reducing ends. Correct? Remember, glycogen molecule is broken down, cleaved from non-reducing end one by one. Okay? By the enzyme. The name of the enzyme is this. Okay? Name of the enzyme is only this, phosphorylase. Okay? But some books, in order to keep it more simple and more pertaining with glycogen breakdown, add a prefix. If you write only phosphorylase, that is more correct than if you write glycogen phosphorylase. Fine. So what does it do? It cleaves one molecule of glucose molecule at a time from the non-reducing end. How? This reaction. It will release glucose as glucose 1-phosphate. So the reaction will be how? This one. Glycogen with N+1 residues broken down in presence of Pi. Phosphorylase, P. It requires phosphate. Remember, Pi is required. So it forms glucose 1-phosphate and glycogen with N residues, removal of one residue. Correct? Glycogen N+1 residue we start with, then we get glycogen N residues and one glucose molecule in the form of glucose 1-phosphate. Enzyme is phosphorylase, which requires inorganic phosphate. অন করো কম্পিউটারগুলোকে, কম্পিউটারগুলোকে অন কর, এটাকে অন কর, রিমোটটা দিয়ে। হ্যাঁ, এসছে, এবার আসবে। Follow? But now the problem is this particular phosphorylase enzyme cannot go on cleaving all the glucose molecules like that. It stops 4 residues away from the branch point. What is a branch point? This is a branch point. This is 1,6 glycosidic linkage. These are all 1,4. So 1,4 they can cleave, but not beyond this. 1, 2, 3, 4. It stops beyond or 4 residues away from the branch point. It cannot cleave beyond that. কি বললাম? 4টে রেসিডিউ আগে দাঁড়িয়ে যায়, এরপরে সে কাটতে পারে না। We need a separate enzyme for that purpose called debranching enzyme. Debranching enzyme. Debranching enzyme is an enzyme containing two enzymatic activity. It will transfer these residues from here to here. It will transfer 3 to 4 residues from one chain to the neighboring chain. This is transferase activity. So it is called alpha 1414 glucan transferase activity. And in the process, it also cleaves this particular glucose molecule, which is different from other glucose molecules here. Why? It has two linkages, alpha 16 and 14 both. That branch point glucose is removed by the same enzyme, by alpha 16 glucosidase activity. So debranching enzyme has got two activities. One is alpha 1414 glucan transferase activity and alpha 16 glucosidase activity. So the process produces a very long chain. Okay, so these three will come here, correct? And these particular process, these glucose will be released in the form of free glucose. So the reaction will be like this. See the color code. One, two, three, four. Correct? It will cut up to here. Then what will happen? These three will be transferred in the neighboring by name of the enzyme. It has two activities. Correct? Then this green one is released as free glucose. Green one is released পড়ো এবং লেখো, পড়ো এবং লেখো। ছবিটা এইভাবেই আঁকতে হবে যেভাবে দেওয়া আছে, ছবিটা এইভাবেই আঁকতে হবে। আবার যাচ্ছি আমি এখান থেকে ছবিটা আঁকা শুরু করো। স্টার্ট ড্রয়িং ফ্রম হিয়ার কুইকলি। 3 রকম কালার দেবে বা ডিজাইন করবে যাতে বোঝা যায়, তাই না? তাড়াতাড়ি তাড়াতাড়ি, হাত চালিয়ে হাত চালিয়ে হাত চালিয়ে, ফাস্টার। তাড়াতাড়ি আঁকো। ব্রাঞ্চ পয়েন্টের থেকে 4টে স্টেশন দূরে, 4টে ইউনিট দূরে ওটা আটকে যাবে এইখান পর্যন্ত। এই ব্লু গুলো আছে, ব্লু কালার গুলো, এগুলি প্রত্যেকবার একটা একটা করে কাটবে বাই ফসফরিলেজ, তাই না, বাই প্রডিউসিং গ্লুকোজ 1-ফসফেট, রাইট? সেই গ্লুকোজ 1-ফসফেট কি হবে, সেটা আবার গ্লুকোজ 6-ফসফেট কনভার্ট হবে, পরে আসছি। কিন্তু এর পরে যেটা হবে, it will remain like this, because phosphorylase cannot cleave beyond this limit. So debranching enzyme will come into action. It has two activity. One is transferase activity of 1,4-glycosidic bond. Another is glucosidase activity. So it will not only transfer the remaining 3 residues from one chain to the neighboring chain, it will also cleave the green colored dot glucose as free glucose. So muscle glycogen breakdown, if the question comes like this, muscle glycogen breakdown can produce small amount of glucose. Explain. How do you explain? Because muscle has no enzyme called glucose 6-phosphatase. Glucose 6-phosphatase enzyme is absent in muscle. That is why muscle glycogen breakdown cannot contribute to blood glucose level. Muscle has no glucose 6-phosphatase activity. Hence, muscle cannot contribute to blood glucose level by breakdown of glycogen. But by the process of debranching enzyme, one molecule of glucose can be generated per at least 8 molecules of glucose 1-phosphate, because the maximum frequency of branching for glycogen is after every 8 residue. So after every 8 glucose 1-phosphate molecule, there is a possibility 1 molecule of free glucose can be generated. So this line is valid. This is a minor fraction of glucose released from glycogen during glycogenolysis in muscle. Right? This is the explanation of production of small amount of glucose, free glucose from muscle. But that is too little for our whole body requirement. That's why we do not count it. Rather, we must rely on glycogenolysis of liver, because liver has glucose 6-phosphatase enzyme. Try to remember these enzymes. These are very, very important. I tell you, the carbohydrate metabolism is most complicated metabolism. Lipid is the most vast, and protein is most discrete. Everything is separate from one another. There is nothing common reaction. Lipid is vast and carbohydrate is very, very complicated. If you do not know the effect of one particular hormone on carbohydrate metabolism, do not try to intimate it. Like think like conceive or perceive that it will happen like this. Actually, it will happen oppositely. Always. Whatever you will think, that is wrong. Whatever you read and remember, that is correct. You try anytime. Challenge is accepted. Whenever you will find, you will think that due to this will happen, you will find that is wrong, and that is why question has been given. to make you read. You must read. The things are different in biological system. You cannot anticipate. This is not chemistry, name reaction, like aldol condensation. No. Things will act differently. Fine. So up to here is done. Now, in the next step, glucose 1-phosphate is converted to glucose 6-phosphate by the enzyme PGM, phosphoglucomutase. Phosphoglucomutase. This particular enzyme requires magnesium ion as cofactor. Okay, phosphoglucomutase, glucose 1-phosphate to glucose 6-phosphate. Done. Now, if it is occurring in liver, then we have the next step, glucose 6-phosphatase. If it is occurring in the muscle, then we will stop here. Okay? See? If it is taking place in muscle, liver, then glucose 6-phosphate will be converted to glucose by glucose 6-phosphatase. If it is in muscle, then we will stop up to here. Clear? Now synthesis. How do you synthesize? Again, liver and muscle are different. So breakdown is different, liver and muscle. See, what I taught you is very small nutshell. I skipped many important, many other points also. So don't think the glycogen breakdown is so simple and so short. Okay? Because of my efficiency, I made it short. You will find it more complicated in your book. So you remember 3 enzymes of glycogen breakdown. What is that? Phosphorylase, debranching, and PGM, phosphoglucomutase, and if it is liver, then glucose 6-phosphatase. If it is muscle, it is. That's all, nothing else. You stop here. If it is muscle, then up to glucose 6-phosphate. If it is liver, then up to free glucose. What else to remember? Remember, in the muscle, what is the predominant metal ion other than sodium and potassium? Yes. So calcium has a big role in glycogen breakdown. When do we need to break down calcium glycogen? When we need energy or when we do not need energy? We need energy. So when do you need energy in muscle? Don't have exercise, Cori. So when we do exercise, what changes is brought into the muscle milieu inside? Calcium level is increased or decreased? কোথায় কমবে? ব্লাডে? মাসেলের গ্লাইকোজেন ক্যালসিয়াম লেভেল কমবে না বাড়বে যখন এক্সারসাইজ করবি? কখন কমবে? যখন তুই পুকুরে স্নান করবি? নর্মাল মানে? মানে রেস্টিং ফেজে যখন থাকবে আর কি। তখন কি হবে? তখন একটু তুলনামূলক ক্যালসিয়াম কোথায় থাকবে তখন? ক্যালসিয়াম কোথায় থাকে তখন? পড়েছিস? মাসেল পড়া হয়েছে কন্ট্রাকশন? বস। So actually calcium will exit the inner part of the layer of the cell in case of contraction. That will trigger the contraction period actually process. So calcium concentration will increase in the cytosol. Okay? The cytosol, that calcium will actually trigger the glycogen phosphorylase, sorry, only phosphorylase, more active. Okay? Moreover, we have effect of insulin and glucagon also on these, that we will study in detail. Before that, we will study the steps of glycogen synthesis, that is glycogenesis. So glycogen breakdown has got how many enzymes? Phosphorylase, debranching, phosphoglucomutase, glucose 6 phosphatase. Just repeat one more time. Good. Then we come to glycogen synthesis or glycogenesis. Glycogenesis, first enzyme is glycogen synthase. Second enzyme is branching enzyme. Lake na. Am I so handsome, good looking, that you are forgetting to write in the examination, sorry, in the classroom? Huh? Don't oil me in such manner. Do not oil me in that way. Buttering has a limit. Buttering has a limit, Paul. You should learn from her. So glycogenesis has got enzymes, key enzymes? Glycogen synthase. Are shon bhalo kore ki bolchi. Listen first. Use your ears first. Glycogen synthase, branching enzyme, phosphoglucomutase, correct? This is enzyme. Glycogen synthase, phosphoglucomutase and branching enzyme. These three. Hello? Now, before we start glycogen synthesis, this reaction is actually not a part of glycogen synthesis, but this reaction is important. This reaction is not a part of glycogen synthesis as such, but this is important. যেমন কুরুক্ষেত্রের যুদ্ধে কৃষ্ণ অস্ত্র ধরেনি, কিন্তু কৃষ্ণ বাংলা বুঝিস? আচ্ছা, কিন্তু কৃষ্ণ না থাকলে যুদ্ধ হতো? অর্জুন বাঁচত? কি মনে হয় তোর? বাঁচত না। তাহলে এ হচ্ছে সেই কৃষ্ণ। কৃষ্ণ কাজ কি ছিল? অর্জুনকে ক্যারি করে নিয়ে যাওয়া। এখানে কৃষ্ণকে অর্জুনকে, তাহলে অর্জুনকে ক্যারি করছে কে? ইউরিন। তাহলে ইউরিডিন হচ্ছে একটা ক্যারিয়ার ফর গ্লুকোজ মলিকুল, তাই না? গ্লাইকোজেন চিনবে কি করে যে কোন গ্লুকোজটাকে গ্লাইকোজেনে অ্যাড করতে হবে, তাই না? কোন গ্লুকোজটাকে গ্লাইকোজেনে অ্যাড করতে হবে গ্লাইকোজেন চিনবে কি করে, তাই না? কুরুক্ষেত্রে একটা গল্প জানিস তো, একজন লোক ছিল যে কৃষ্ণের মতন ড্রেস আপ করে থাকতো। গল্পে আছে, মহাভারতে আছে গল্পে। হ্যাঁ, একদম কৃষ্ণের মতন সে থাকতো আর কি। কৃষ্ণের কোনো কিছু না, তাহলে কৃষ্ণকে নকল করতো আর কি। তো সেরকম ওই নকল কৃষ্ণ না আসল কৃষ্ণ কি করে বুঝবে? যদি অর্জুন থাকে তাহলে আসল কৃষ্ণ, অর্জুন না থাকে নকল কৃষ্ণ। কোথায় বুঝতেছিস? লোকে তো কৃষ্ণকে নকল করবে, অর্জুনকে সবাই নকল করবে। বড়কে, হ্যাঁ, তাই না? তাই কৃষ্ণকে নকল করেছে, কিন্তু অর্জুন তো সঙ্গে নেই, তাই লোকে বুঝে গেছে আরে এ তো হবে না, এ তো আসল কৃষ্ণ নয়, সঙ্গে অর্জুন নেই। বুঝে গেছে? তাহলে কোন গ্লুকোজটা গ্লাইকোজেনে ঢোকানো হবে কি করে বুঝবো? If it is attached with UDP. So UDP is actually a carrier for glucose molecule. If you recall, the function of nucleotides acts as a carrier molecule. CDP choline carries choline. UDP glucuronic acid carries glucuronic acid. Glucuronic acid কোথায় লাগে? In the synthesis of mucopolysaccharide, ionic acid pathway, right? Similarly, here UDP glucose. So this synthesis process is catalyzed by this enzyme. Read the enzyme. Read it. Ponna, Jojo Ponna. UDP glucose pyrophosphorylase. আবার আমাদের দেখাচ্ছে না, আমি লজ্জা পেয়ে যাবো এবার। UDP glucose pyrophosphorylase, UDP glucose pyrophosphorylase। আগে এনজাইমটা কি ছিল? phosphorylase খালি এখানে কি আছে pyrophosphorylase that means what phosphorylase means addition of one phosphate pyrophosphorylase means addition of transfer of two phosphate pyrophosphate means two phosphate so it will transfer two phosphate from where this is glucose 1 phosphate see the phosphate in red color now UTP uridine plus 1 2 3 uridine triphosphate correct এর মধ্যে দেখো only one is blue color rest all black this is called alpha phosphate this is called beta phosphate this is called gamma phosphate so beta and gamma phosphate are removed only alpha phosphate is attached with the red color glucose 1 phosphate huh? See red and blue and black is removed since these two phosphate are being removed by this enzyme it is called pyrophosphorylase in breakdown glycogen breakdown it was phosphorylase because it was removing only one phosphate here it is removing two phosphates so it is pyrophosphorylase why this particular step is important in most of the reaction like hexokinase glucokinase PFK1 PFK2 we have seen or pyruvate kinase we have seen transfer of phosphate from nucleotide here we are seeing the opposite transfer of nucleoside not phosphate what is important here we are attaching one nucleoside to the glucose right not the phosphate we are removing so this is the other way of transferring phosphate than kinases আমি 1 ঘন্টা পরে করি ক্লাসে আছি এই কটা বাজে রে যাব বাবা বলবি তো fine acha so write down first this session is called activation of glucose this step is called activation of glucose লেখা আছে এটা লেখা আছে তো লেখা আছে রিঅ্যাকশন অ্যাক্টিভেশন অফ গ্লুকোজ হলো এবার পরের স্টেপে এগুলি দরকার নেই এগুলি বেশি ডিপে কি লাভ নেই পরের স্টেপে যেটা মনে রাখতে হবে আমরা অ্যাড করব we are going to add one molecule of glucose at a time but to what to a small molecule called glycogen primer now a new term has come glycogen primer what is glycogen primer we will discuss in next class